So we talked about different types of motif representations. We talked about how to find motifs in sequence data. And essentially we're dealing with collections of words here that we're matching against some kind of corpora. Now the most interesting aspect to this would be the discovery of these sort of sequences of words. And that's what we're going to spend time on talking about today. And we're going to develop the intuition behind one particular form of discovery, and that's through so-called Gibbs sampling. That's an example of the latter type of method here, which I call discovery by model, one size fits all. So we're going to find a representation of a subsequence. And this could be a profile like the one that we talked about on Tuesday. And that is a profile that maximizes some objective that we define in terms of the data that we have accessible to or access to. The other approach would be by enumeration, and I'll finish off talking about that once we have an understanding of what this more sort of elaborate approach involves. Enumeration is simply where we think about so we have our data, we have some kind of model, I'll refer to it as theta. So we define our model in terms of theta, and this approach here would find theta that would maximize its ability to reproduce what we see in the data. The alternative would be this one here, where we actually look at all data points and we find something that is enriched in that data and subsequently find our parameters. But we'll look at that alternative later. Okay. So there is this idea behind quite a few different discovery methods, and we'll be thinking in terms of, we'll define our problem in terms of two variables, or two sets of variables, if you like. One that I'll be referring to as Q, which is our model. You can think about it as the profile that we talked about on Tuesday. The other one is a set of labels. Essentially, some way of labeling our data so that we can form our model. So this could be, say, a position in a sequence that we regard as a match. So a discovery problem would then be to find the values for Q and Z, or Z, that maximize this joint probability. So on Tuesday I talked about joint probability as being one that could be thought of as a big table that enumerated or listed all possible combinations of the value settings of these variables. We'd like to find the entry that has the largest value, and that would, or the largest probability, and that would be the one which identifies what our profile would look like and what our labels would look like. So what the motif looks like, as well as where the matches are in our sequences. The problem is that we don't know those values. We're actually very information poor. We have neither Q or Z here. So we take a different approach, and we'll be using something called expectation maximization or Gibbs sampling to do roughly the same thing. Two things. From the labels we can find a profile. So once we know the labels, so we have some kind of classification, if you like, or categorization of our data, we should be able to use that in order to find our parameters. Okay? We don't have those labels, but let's just pretend for a moment that we do. The other way to think about it is that with a profile we can find the labels or the categories. That is, once we have this, we can identify where our matches are. Of course we don't have that, but if we pretend that we do, then we would be able to label these things. So those are the two perspectives that we'll be using. So when it comes to motif matching, and I'll use that as the penultimate example, but I'll use a simpler example before we get there. We have a set of sequences, and let's pretend that we have the labeling of them. That means that we would have the positions at which we find our sort of special pattern. So they're all aligned here, so this red bit lines up very well. And from there, we know how to calculate this profile. We talked about that on Tuesday. And that would include having some kind of background distribution, and we'll sort of collectively refer to this as Q, our set of parameters that define the model. Does that make sense? So that was the first step that we talked on the previous slide. The alternative is this one, where we pretend that we have this, and we identify where in the sequences that we have our match. And in the simplest form at least, we make assumptions about how those labels can, or how they are constrained. So the form of motif discovery that we'll discuss assume that we have exactly one match in every sequence that we have. You can come up with refinements or variants of those, such a method, to accommodate the fact that in some sequences you have no match, and in some sequences you have two or more. Either way. Okay, so those are the two very important perspectives, and if I flick back, we realize we don't have this labeling, but if we assume we just randomly assign these labels, we would be able to compute a profile. It wouldn't be particularly good, at least in the first instance, but let's just do that. We go through the exercise, and then we use that particular profile in order to predict the positions in the sequences. Now, you would normally assume then that, well, if we do that, well, we should get the same positions, right? Okay? But it could be that there is a better match for one of the sequences that could, so even though we identify a position here and use that to compute this, it might be that the others are completely different and dominate thus the profile that we compute, so that we would prefer a different position in that very sequence. That's the whole idea. And there are two classes of methods that will deal with this problem, and one is expectation maximization, which is used for many different types of problems. So not only motif finding. It finds the values for Q and Z or Z that maximize this joint probability given the data, and this is a joint probability distribution that we don't know explicitly. Okay? So we can't simply learn it. We have to use some kind of iterative approach that would explore the space of possible parameter settings. So EM does this by iterating between two different steps, and one is the E steps, and you can probably guess that corresponds to expectation here. So this is the expectation step. We estimate this thing here, so the probability of the labels given our model. So that would be when we take our model and we make predictions of where the matches are in the sequences. And then we have the M step, where we do the opposite. We take our labels and we estimate our model, or the parameters of the model. And just doing this iteratively will actually allow us to approximate this joint probability here. These two sets of variables will continually or gradually improve as we go through these steps. So we repeat until this change in the model parameters is very small, so we don't improve anymore. This is a deterministic method and will converge, but it is liable to get stuck in local minima or local optima. That is, that we might have a solution somewhere, but it might not be the best one we can find. Okay, so the one that I'll focus on is Gibbs sampling because it's slightly easier to understand. But they do share quite a few features, and they can be used to solve the same type of problems. So Gibbs sampling samples, as the name suggests, from a joint probability distribution not known explicitly. So the same kind of problem, it's just that EM doesn't sample. It establishes the beliefs in each of the values that we predict. Whereas this one here will sample this joint probability distribution, and we'll see how in a moment. So Gibbs sampling is a stochastic analog of expectation maximization. It repeatedly samples from these two conditional probabilities. That is the probability of the model given the labels and the probability of the labels given the model. Now, what we'll do in order to truly sample this, we have to withhold one sample or more samples. Otherwise, we'd be sampling training data, and that wouldn't be a good indicator. So we're going to withhold a data point, and I'm going to illustrate this with a simple example shortly. And it's the same thing as with EM, that Q and Z will improve gradually, but in this case it's done stochastically. So we could have jumps or actual, we have changes that might decrease performance, but that would be just occasionally. Most of the time we would have improvements. Because of this stochasticity, it's less susceptible to get stuck in local optima, so it's therefore preferred in some problems, but essentially they're doing the same thing. Okay, so we're going to go through an example where we find Q, which will in this case not be a motif profile, it will be just means of two Gaussians. And we're going to find Z, which would correspond to labels of data points. So we're going to have a very simple problem. I'm going to do some drawing on the whiteboard. So here's our data, or our data axis, and we would have a number of different data points. And I've indicated them like that. And if we were to find the most probable, so we have let's not do that. If we want to have the, so each of these would be labeled, so they would belong to two different classes. And I'm going to represent those two classes with a Gaussian each. So I can draw a Gaussian, might look like that, and another one might look like that. Now I might refer to this one as A and this one as B. Now, depending on where I put these, I have an opportunity here to maximize the probability of generating that data using those Gaussians. Okay, so if I were to generate data from A and from B now, most of my points would end up here, right? And some over here. So if I wanted to reproduce the data that I have here, I would probably want to shift this one here, so it's over these guys, and then maybe modify B slightly too, so I would have that over those guys over there. That would be the optimal setting of those two Gaussians in order to maximize the ability of them generating the data that I have. Does that make sense? So I'd like to have a method that finds the parameters of those two Gaussians. And I'm going to use Gibbs sampling to find that. So I have a couple of slides with lots of numbers on them. And I apologize not for having the actual graphs here, but I didn't have access to a good plotting program when I was generating this. So I have 8 different data points, x0 up to x7. And I've just assigned them different values. So the smallest one is 2.9, the biggest one is 8.8. Now, we said that we would be, we don't know the labels of these. We'd like to find the Gaussians that will give us, or we'd like to find the joint, the maximum joint probability of the Gaussian parameters as well as the labels. So the labels that give us the most probable, the Gaussians that would most likely generate or regenerate our data. I start off with just randomly assigning labels to my data points. Yes?
A question. You said that you skip your local. Are you using a high momentum?
So there is no momentum here. So this is not like a neural network, if that's what you're alluding to.
And for the Gaussian, are you using oversampling or something like that? Push the Gaussian up or not?
Do you mean when you say push up the Gaussian, you mean that I use different variants, or?
Yes, exactly.
Yes. So in my example, and it's good that you asked the question, I'll simplify matters a lot. There would be, obviously you can set your variance so that it's as tight as possible over the data points, because that would be the way to maximize the probability of regenerating those particular points. But in this case, I'm going to assume that we have a constant variance. I will stick to 1 as a variance, but we could equally well incorporate the variance as a parameter of the model here. So the only thing that we're looking for are the two means of those two Gaussians. But this is just to illustrate the idea behind it. So we start off with just random assignment of these labels here. There are 2 steps. So we have the predictive update step. We update the model, Q, given the current labeling. And then we have the sampling step. We sample a new labeling given the model that we just updated. And then we're going to repeat those 2 steps. So those are exactly the same that I talked about earlier.
I'm really confused again. So these are the labels that the X are the values that you draw.
These are the
X on the whiteboard.
So
right?
X.
What is Z?
So Z is the label that I assign to each of them. So that would be
distribution 1 or distribution 2, A, B. In your example on the whiteboard.
Yes. So we can call this 0 and this 1. Yep. Good point.
So we start with an initial labeling?
Yes.
So this could be one of the initials and then the 3.
Yeah. So we could, if we start here, now normally you would assign the labels randomly with Gibbs sampling, and then you determine the best Gaussians given those labels, right? That's what we're going to do. Okay, so we're going to update the model here. So we start with that random assignment of labels. We work out the best Q. Okay, so now the important thing here is that, as I alluded to earlier, we're going to withhold 1 data point. That's the one that we're going to sample later. So I'm going to disregard X4 here. It's 6.4. We're not going to look at it.
You need 1 per label?
No.
Just 1.
Just 1. So, and then I would have my random labels initially. They would either be 0 or 1, or A or B. Okay, doesn't matter. So in this example here we have 0 and 1. So we have these 2 that ended up in class 0, or with the label 0, 2.9 and 6.1. From them we can determine the parameters of our model. Or one part of the model, I should say. This is the mean of one of the 2 Gaussians. I can determine that. It's 4.5. And I just keep that constant there. But we could estimate that as well. And then we have the samples that ended up in the other group. And these are the ones. And we determine the mean and potentially the variance for that set. Okay, so now we've gone from our labels to our model parameters. And we use those model parameters to determine. And now we're going to look at those two Gaussians and see where our withheld data point x4 ends up. So, and that's going to be a probability distribution. So we're going to say we're not quite sure whether the label should be a 0 or whether it should be a 1. So if you look at these here, you can determine which one of those two densities are the biggest. Actually, when you determine the posterior, you're going to end up with something where the probability of 0 is going to be 0.5 all the way up to where these two cross, if the variance is the same in those two. Otherwise it's slightly trickier, but that's where our posterior probability ends up. And that will give us sort of a distribution from which we can sample. So we say, well, we're going to say that x4 is now going to be labeled 1. And we came to that conclusion because we randomly sampled that distribution, and because this is bigger, there is a greater chance that we pick 1 as opposed to 0. But we could pick 0. That's the stochastic element of Gibbs sampling. So sometimes, if we're unlucky, we're going to pick that one. Well, I'm saying unlucky. I don't know that, because we might be in a local optima and we want to get out of there. And we just take a penalty or a decrease in overall accuracy by maybe picking the class 0 there. But that's the whole idea. Does that make sense? So now we have a label for x4, which I'm going to add to the mix. Okay? But at the same time as I add it to class 1, there it is, I'm going to withhold another one. So I'm withholding x1, which was previously, if I flick back, in class 1 as well. But that's just randomly selected out of all the samples that I have. So again, I do the same thing with the Gaussian distribution that corresponds to the label z equals 0. Of course, these are the same, so the result will be the same. I do the same thing with the other Gaussian. I get to a slightly different mean, which means that I now have a new basis in order to see whether I should be relabeling x1. I use the two Gaussians, look at the posterior, I get this distribution. So clearly I should be picking class 0 here. It's pretty clear. Although this is probably not exactly 1, but very close to it. And that's what I do. I end up sampling, get label 0 for x1. So that's what I get in the next time step, in the next iteration. So it ends up over there. Same process. This time I'm holding out x3, and this is the distribution I'm sampling from, and I pick 1. And there it is. I hold out x2 and do the same computation once again. And at this stage, I think we're almost done. Now, this will continue until we see that there is no improvement of the model parameters, or that there is no change in the model parameters, and that's when we're happy. And this is the final classification, or the final labeling of those data points. Okay. Did you get the principle? Okay, so let's apply it to motif discovery, which is slightly more complicated because we have a more complex labeling scheme. You remember that we wanted to do this kind of alignment. So we're given n sequences. This should be a big number. ideally. We seek within each sequence mutually similar segments of a specified width. So our search will be for a particular word length. And we initialize the parameters, and this would be the alignment. A position in each sequence. So here's where we make the assumption that there will be exactly one match in every sequence that we have. This is something that we may consider relaxing if we have a problem where we just have a bunch of sequences and where we expect there to be a subset that have one or more matches. But let's just pretend that we know that there is exactly one position in each sequence that has a match, and that will help us to develop a fairly straightforward method. So we have the same steps, so predictive update step, where we update the profile in this case rather than the means of the two Gaussians. This is our Q, given the current alignment. And then we have the sampling step where we sample a new alignment, sort of find new labels given our profile. Sorry about the color scheme here. Somehow when I copied between different PowerPoint shows, the color scheme went berserk. So, but I hope that you can read this. So we have a motif length, W, instead of n sequences. We have two data structures that we need to maintain through the search. We're going to actually include yet another position in our profile. So we're not just going to store our W distribute. So we would have our motif, which would be distributions for every position in the word. So we would have position 1, 2, 3, 4, 5. We would have probability distributions for each of them. So the different letters that might occur in those positions. And then we would have an additional parameter that we'll refer to as P, which will just be a single distribution. And that would be the background from which we like to distinguish what's the real and just the background. Okay, so we have the actual the matrix here, Q, and we have a background distribution. So those are the two things that our profile consists of. Then we're going to have our labels, and these are the start positions of the pattern in each of the sequences, and I'll be referring to them as A. And K here is an index for the specific sequence that we're looking at. Okay, so you don't have to sort of look too closely at the indices here or think too much about it. You'll have access to the slides if you want to go through this in detail. But I just want you to go away and have a feel for what this does rather than having sort of just understood everything perfectly. Okay, so the first step is to initialize. Just like we did with the Gaussians, we labeled our data. So we're going to start with our word length and a random set of locations referred to as a index K. So we're going to randomly assign locations in our sequences like this. So our labels here are going to be a position in each of the sequences. A start position. Yeah. So first up, the predictive updating step, where we form our profile. We select the sequence at random. I'm going to pick this one here. And then we're going to determine a profile on basis of the black sequences here. And we're going to look at two things. We're going to look at the counts in the orange sections here, which will help us to form this. And then we're going to look at the counts in the black section to form the background. And there are a number of different variables involved here. So this is the formula. What you need to know, or what you should just note at this stage, and then you can look closer later, is the first two, or this fraction here at the left side. So we have the count of a particular symbol at a particular position relative the start position of the alignment, and then we have the number of sequences. So we look, say, at the first symbol that occurs in each of these positions, and we're going to be able to determine a frequency or a probability of each of the letters in that particular position. The rest here is just something that we refer to as pseudo counts. It gives us an ability to deal with the scenarios when we have zero counts of a particular letter, and we still want to allow the profile to assign a small probability to their occurrence. This works really well when we have small data sets. It helps us a lot. But you can disregard it for the time being, just to see the main principles here. Okay, so we take that away, we do the counting, and then we're going to do the counting necessary for the background. So this one first, now this one. So we look at all the black sections here, and we count the letters, and now we don't care about the positions anymore. And we find a number for that as well. Now we get to the sampling of the alignment step. We're going to adjust it in the selected sequence. So now we bring back our withheld sequence, and we're going to score it. So we're going to generate a histogram. This is actually a probability distribution because we're going to normalize it so it adds up to 1. And we're going to assign a score to each of the positions here, which indicates the fit to this profile, which is there, divided by our background. So it's a ratio between the two. So if we have a big probability assigned by the profile, so a good match to this, and a poor match to the background, obviously this will be a big number, probably some of these guys over there. Alternatively, if we have something that doesn't look properly like the profile, but instead matches the background quite well, we'll have a low score. And we do this for every single position. So we're sliding this sliding this profile, or this matrix here, across the whole sequence. This was the previous assignment, which we disregard completely. And we're going to sample. Now this is the sampling step. You remember where we said, well, that for the two Gaussian examples, that we assign a new label, one of the two, 0 or 1, depending on which of the two Gaussians that we're sort of assigning it. Or we sample those two Gaussians, the probability distribution that we can generate from those Gaussians, to find the new label for the data point. Similarly, we're going to assign a new start position for this sequence by sampling this. So it's a greater chance that we pick a position like over there, or maybe there. But there is a small chance that we'll pick a start position like that. But that's just a very minor chance. So we accidentally pick that one there, which is quite good. And we start again. So step 3 is really to go back. So go to 1 until convergence on a set of positions. So the procedure is repeated from step 1 until convergence. And initially the selected positions are not more probable than any other. So we have a relatively flat distribution. So when we look at this thing here, it'll be more or less flat. We're going to explore possible positions for our motif. And that's good because we haven't really made our minds up yet what the motif would look like. But eventually, we will converge on something that happens quite a lot in all of those sequences. And convergence is quite likely with this method. So initially, selected positions are not more probable than the others, leading to exploratory updating. Later, correct positions with patterns shared with the other sequences will introduce a bias into updating leading to convergence. So I think that there is some animation here. So we pick one sequence at a time and update its label. That's all that I was going to say about Gibbs sampling. I'm going to say a few more things about probabilistic models in general. And I should add that expectation maximization operates on the same principles, would get into trouble in the same scenarios as Gibbs sampling does, but would also have that additional problem of getting stuck in local minima. But it works relatively well when we face problems that don't have a lot of dependencies between positions. So remember that this probabilistic representation of the motif doesn't really consider dependencies between the positions. We have simplified. So if we think about these as maybe 5 different variables, x1, x2, x3, x4, and x5, we're really looking for a way of computing the probability of all those 5 variables and seeking the one which is highest. Now, normally we'd like to capture, or maybe we'd like to capture dependencies, but if this is a probability distribution across 20 amino acids or 4 nucleic acids or even 2 values like true and false, it's going to be a very big joint probability table. So we make the simplification that we treat each of these columns independently. So what we have done is to say that that's approximately the same as this product, and so on. And that's a problem with probabilistic models, that they tend not to work well when things are very complex or open-ended. In which case we need to introduce what we think are reasonable dependencies. And you've seen hidden Markov models in this course?
Seen, yes, but not really discussed.
Okay. Where we break this or decompose this joint probability into a series of conditional probabilities that could represent dependencies. But even then, complex models would require large data sets. The best publication, if you want to know more about how Gibbs sampling works for motif discovery, was published quite early on. It's really a seminal paper, and it's very easy to read. So I would strongly recommend that you have a look at this. And if you look at that, at the same time you can look in this implementation here, which we developed for a course that we're offering at the University of Queensland. You can see that, this is written on basis of what they discussed in that paper. So you'll see the same notation and so on. So it'll be easy to follow how this could be implemented. So you're welcome to use that if you like. So this is a slide to just illustrate one of the experimental technologies, and then since you're computer science students, I'm not going to bore you with the intricate details of this experimental technique, which allows us to identify sections of DNA that have binding sites that we'd like to create motifs for. Now, I talked about this early on in the lecture we had on Tuesday. This so-called ChIP-seq experiment or experimental technology, which is based on a series of different steps. But what it gives us in the end is really positions on the genome sequence where we see an enrichment of, where it's likely that our protein of interest binds to DNA. In bioinformatics, we like to look at things, and there is a tool called the genome browser. There are a couple of different genome browsers actually. One is available over the internet, the UCSC. genome browser, and you can look at graphs like these. And I'd just like to point to a couple of tracks here, as they're called, which are displayed across the genome sequence. So here we see the different positions, and then we're going to look at a particular experiment, and we have an example of a ChIP-seq experiment at the top here. And that's really the signal, the magnitude of the signal that we see for that location. So here the experimental technology has given us a clear indication that there is something going on here. So we should be looking closer at the sequence at that particular site. And also over there and over here. So we can extract those sequences and run a Motif discovery tool such as the one that I just discussed with Gibbs sampling and see whether we can find the motif. Now if we think that there are dependencies there, or whether we want to try the alternative approach that I started off talking about, namely enumerative motif discovery, where we basically look at our data set and identify all possible subsequences that could form part of such a motif, and then subsequently create a probabilistic representation of it. We need to think about these things here. So on Tuesday we talked a little bit about joint probability, where we have a number of symbols that we'd like to consider jointly, that they appear next to one another, for example. And how do we find them? Well, we can do so by counting. So I'm coming back to the idea that we have access to some kind of big sequence set, and in this case here I have some Python code that refers to a data set that consists of 2,300 sequences. And these are sequences that have been generated by such an experiment that we saw on the previous slide. So there were 2,300 peaks like that, and around each of those peaks, so we would have no. My red pen is probably not very good, so I should switch, right? Okay, so let's remove this. So we had 2,300 sequences, and I'm going to extract 100 symbols around there, or a sequence that is 100 symbols long. So add 2 zeros there. That's how many base pairs, or how many symbols that we're dealing with here. So it's 230,000. If I look for the number of A's I have there, so I'm just looking at one position, so something like P of X, and that X equals A is probability of X equals A is 0.21. And then we have the others there, 0.28, 0.28. We see that C's and G's are slightly more frequent than the other two. And we have reliable assessment of those probabilities because we have big numbers. Now if I want to identify a pair instead, so we're talking a joint probability of X equals A and Y equals A, say. We can estimate that in the same way. We just count. And it would be 9,800 occurrences of A followed by A, which is 4% of all the dinucleotides or dimers in our sequence data. And then we can, and I should note also here that you notice that G and C were the two most frequent letters. And if we look at the two pairs C and G, it's actually one of the least frequent dinucleotides. And then we have G and C, which is one of the more frequent. So clearly the first approach is not going to be able to distinguish between those two tendencies in our data, that they're so different when they occur in that order or in that order. And then we go to 3, and you see that the numbers are starting to drop here. So we have fewer and fewer data points on which we can make an estimate of those probabilities. And now I can't even fit them all on the same page. There are 4 times 4 times 4, so 64 possible assignments here. So clearly we're dealing with bigger numbers. And if I would go to 4 or 5, we need more data. So this is a method by which we can detect dependencies, because we would see the difference between CG and GC, for instance. Whereas if we had a simple zero-order model, we would not be able to do that. That is, that we only look at one nucleotide at a time. Same thing with amino acids. So if we go to a different type of alphabet that would define our corpora, we would have the same kind of issues. And it's even worse with amino acids, since we have 20 different assignments. Okay? So this would now give us, so if we collect these trinucleotides, we would have a model for motifs that would be 3 nucleic acids long, right? Or analogously for any other alphabet. These probabilities would help us, especially if we also do this for a background set, right? What we have here is the skeleton for an enumerative method. And in fact, I'll just outline a very simple approach to take these types of counts and make a statistical assessment of which is the what type of motif is enriched in these sequences. So we're using the ChIP-seq data set here, but this could be anything. But one where we have indications of a hot region, or one region that is likely to be enriched, versus regions that are likely to not be enriched or unlikely to be enriched. So, and this would be our background. When we're dealing with protein sequences, this could be sort of negative or proteins that we know don't have this particular feature, and this could be our positive set where we know that the feature exists. Okay? So we had different sequence sets, blue and red here, but red is positive. And we can make we can design these data sets on basis of our understanding of the domains. In the case of the ChIP-seq data, we know that when we look at those peaks that I showed you earlier, that the binding site is typically within 100 base pairs, or in a 100 base pair window, around there. And now we start counting. So we detect motifs that are enriched in central regions, and we compare those numbers with those in the flanking regions. So we simply count the number of sequences containing a word in the two sets. So we pick all possible words, for instance this one here. CA, CA, CCC. It's just one out of, and it's a length 7. So that would be 4 to the power of 7 possible words to go through and do the same test. So for this one here, we're going to tabulate, as we will do for all the others, how many sequences that doesn't have the word in the negative set. So in this case it would be 606 out of the 2,300 or so. Sorry, that's not the same data set here. So it's a large number of the negative set that don't have the word, and there is a smaller number in the positive set that have that word. Okay, so from that we wouldn't be particularly convinced, I guess, that that word might actually be something that we should be using as part to define a matched motif. But then we look at this column here. So we look at a negative set again, where it has the word. Sorry, I'm not sure whether I framed that correctly here. So the sequence doesn't have the word. So negative set, lots of them don't have the word, but lots of the positives don't have the word as well. And then we look at the numbers where the sequence does have the word, and some of the negatives do. And actually, this number here is larger than it was for the negative set. And if you compare that to the proportion that we saw of sequences that don't have the word, this surprises us. And there is a statistical test for assessing this called the Fisher exact test. And we can get a p-value. This would give us a number from which we can say that this is a good or a bad word to make use of as part of forming a motif. So this is a very small p-value, which means that it's very unlikely. So if we were to generate words just randomly, it would be extremely unlikely that that word would have this kind of proportion that we see in this contingency matrix. And we have to do this for every possible word. And then we identify the top ranking ones, and we would put them together. We could form a motif or a representation of them that could be probabilistic and be used as for scanning new sequences or for possible matches. So that's an example of a enumerative motif method. And I'm almost done. So we have discrete models of sequence composition, and these are sensitive to noise in the sense that we could have so in the same sense as for where we had the probabilistic models, we introduce so-called pseudo-counts in order to make them more robust to the fact that that we haven't seen all possible combinations in our data set. The problem here is that we might have a word which is highly enriched, but if we flip just one or two symbols here, we have a very similar word, which could have a very different word count. And this could be somewhat difficult to use when you form a unified representation of those that have a high count. There is also the obvious problem of space and time, that we have to go through all possible word combinations. We make this assessment by scanning through, in this case, 4 to the power of 7 possible words. That could take considerable time to do, and if we go to even longer words, the increase in complexity is exponential. There is what I think is a great suite of tools known as the MIME suite, which contains motif discovery methods for the two types of approaches that we've discussed here today. So MIME would be the method that uses EM, or expectation maximization, to produce a probabilistic model. DREAM would be an alternative which uses enumerative motif discovery. And you can actually run them both on the same data set quite easily and get results that are usually quite comparable to one another, but they have get there through completely different means. I think that's all. So I think I'll finish there, which is a little bit early, but I think we're okay with that, right?